Okay, so chapter 11. In this chapter, we're going to talk about mechanisms of microbial genetics. So, as you learned in previous chapter, DNA is the genetic material which is responsible for passing all the genetic information from parents to the offsprings, and doesn't matter if you are a bacteria or if you are an elephant, so you will transfer all those genetic information encoded in DNA to your offsprings. So a little review or refreshing about the DNA structure. If you remember, DNA is a macromolecule which is made of a smaller subunit that you know them as nucleotides. So we have 4 different types of nucleotides in DNA. We have adenine, guanine, cytosine, and thymine. And adenine and thymine, they are complementary to each other, and guanine and cytosine, they match each other, or they complementary to each other. And as you know, DNA is made of the DNA backbone is made of phosphate group and a 5 carbon sugar that we know it as deoxyribose sugar. And so you can see it in this picture. So this is the phosphate group and the sugar, and then they make them phosphodiester bond with together, which is like the phosphate gonna make a bond with the 5 prime carbon in the deoxyribose sugar, and then through the hydroxyl group that we have on carbon 3, so it's gonna attach to the next phosphate. So one phosphate is attached to the 5 prime carbon, and the other phosphate is actually attached to the hydroxyl group that is actually connected to the carbon 3 prime carbon in the sugar, deoxyribose sugar. And then so it makes the backbone of the DNA, and those nitrogenous bases that we talk about, so they make actually, they are in the inner side of the DNA. And then if you imagine like a ladder, so the backbone is made of sugar and phosphate, so phosphate group, so you can see it like here. And then we have a 5 carbon sugar, so you can see it like this. And then the inner layer is actually made of nitrogenous bases that you learn about, like adenine with the thymine, and cytosine with guanine. So they make like a hydrogen bonds between these nitrogenous bases or nucleotides, and then they make the structure of the DNA as a molecule that you know it as helical double-stranded. So it's a double-stranded, it has 2 strands and it's helical, and these strands are complementary to each other and they are anti-parallel, so it means they run in opposite and in different directions. One strand is from 5 prime to 3 prime, the other strand is 3 prime to 5 prime, and phosphate group is at you can see the is at the 5 prime ends, and the sugar group is at the 3 prime end. So now you know more about the DNA structure. Let's talk about some of the differences in the DNA structure of prokaryotic and eukaryotic cells. So prokaryotics, they usually have like one large circular chromosome. So they usually have 1 or 2 chromosomes, for example, in bacteria. And their chromosome is circular. So you can see it in this picture. So this is like a chromosomal DNA that they have. And also they have some smaller DNA that you know them as plasmids, or they are small circular DNA that they can carry some important genes such as antibiotic resistance genes or production of toxins. So these genes are stored in the plasmids, and they don't have any nucleus, so that's why their DNA is located in an area that we call it nucleoid, that doesn't have any membrane or any membrane or boundary around this nucleoid. So but this is the region that the DNA is located. And eukaryotic cells are much more complex, so they have usually multiple linear chromosomes. For example, in human cells, we have 46 chromosomes or 23 pairs, and they are very supercoiled, so they are really twisted around a protein that we know it as histones, and then they are located in a membrane-bound organelle that you know it as nucleus. So doesn't matter if you are a bacteria or if you are a human cells. So DNA has two important functions in all the living organisms. The first role of the DNA is the inheritance. So DNA can pass down all those genetic information from parents to the offsprings, and it's very important to make sure to keep the accurate and precise details and information and pass it to the new molecules and new cells. So that's why we're going to talk about the DNA replication and how the DNA can make a copy of itself with keeping all the exact information without any mistakes. And then it can roll as a blueprint, so it can, because DNA has all the genetic information to make you or to make a bacteria. So it's very important to be able to transcribe first those genetic information to a smaller macromolecule that we know them as RNA, or you're going to learn about it, we call it mRNA or messenger RNA, and then we're going to transfer this information or translate this information to another macromolecule that you know them as proteins, and then we're going to talk about transcription and translation, that the genetic information can actually convert to the functional proteins such as hormones, enzymes, structural proteins, or a lot of different kinds of proteins in your cells. So let's talk about central dogma. So central dogma states that DNA encodes messenger RNA, that we know it as transcription, and then the RNA gonna get translated, and it's gonna turn into proteins, and you know proteins can do almost everything. So we call this central dogma. So genotype and phenotype. So genotype are all the genetic information you have in the cell, and of course you cannot see the genetic information of the organisms, but phenotype is actually, that is like what is expressed. So after the genes are transcribed and translated, so with those proteins that are produced, so then they can give some phenotypic characteristics to the organism that we can see them. For example, the color, your hair color, your skin color. So these are all phenotypes. So genotype gonna stay the same. So but the phenotypic characteristics or the phenotype based on the environment and the condition can be expressed or can be suppressed. So for example, this is a very good example that we have in the lab. Serratia Martians. So this bacteria, if you usually put it at 37 degrees Celsius, so you can see it as like a kind of milky or white colonies, but as soon as you move it to 28 degrees Celsius, so it's gonna produce some pigments, some very interesting pinkish pigments that it couldn't produce it when it was at 37 degrees Celsius. So at a certain environment, so the phenotypic characteristics or the phenotype of this bacteria can changes. So like for example, the color of the colonies in this case. So now you learned about some of the basic information in genetics. So let's talk about the first and very important job of the DNA, which is the DNA replication. So DNA replication always starts from a region that we call it the origin of replication, or ORIs, and so this is like the part that the DNA, which is a double-stranded molecule, gonna get separated. So it's gonna make a replication. So it's gonna make the replication bubble, or that the two openings, they call it replication forks. And then the proteins and enzymes that we're gonna learn about them, they can attach to this region. And then in prokaryotic cells, so this replication can be continued, or can go from the origin of the replication until the entire molecule is actually copied. So some of the enzymes that you need to know, that they have very important roles in the DNA replication in bacteria or in prokaryotic cells. First one is gyrase, or they call it topoisomerase 2. So the main function of this enzyme is unwinding the DNA. So it's gonna make sure that the DNA is not folded and every part of the DNA is accessible to other enzymes. Then we have the helicase. So helicase acts like scissors. So it's going to separate and break down the hydrogen bonds between the two strands of the DNA, and it's gonna unzip the DNA. And then single-stranded binding proteins, they're gonna attach to the each strands, or to the single strands, in order to make sure they are not gonna go back, or they are not gonna back together. And DNA polymerase 3, which is a very important enzyme, so it's going to add the new nucleotide, so that you can see it in this picture, to the 3' end. So we always add nucleotide to the 3'. So if this is our template, 3' to 5', then the new strand gonna be 5' to 3', and we always add the only part that we can add new nucleotide is the 3', because it has a free hydroxyl group that can make bonds with the next nucleotide. And another important part of the DNA replication, so that you can see it in like orangish color, we call this primer. So primer are very small RNAs, or RNA molecules, that we need them in order to be able to continue, or make the new strands with the help of the DNA polymerase. And the enzyme that actually make primers, we call it primase. So primase is the enzyme that can make primers. So primers make primers, and these are small sequence of the RNA that the DNA, with the help of them, the DNA polymerase 3, can get started. So this is a very nice picture from your textbook. So you can see the the different enzymes that we talk about: helicase, type isomerase, single-stranded binding proteins, DNA polymerase. So the important thing that you need to know here, that as I mentioned before, DNA polymerase 3 always makes the new strand or gonna move from 5 prime to 3 prime direction. And if you remember, the DNA is the anti-parallel molecule, so one strand is from 3 prime to 5 prime and the other strand is from 5 prime to 3 prime. Since everything like the DNA polymerase in like in this strand, so it's gonna move this way, and all the enzymes they're gonna move this way, so we're gonna continuously make one of the new strands of the uh, DNA that we want to make a copy, that we call it leading strand. So leading strand, this is the strand of the DNA that we make it continuously. But look at, because the other strand, because it's in the opposite direction, so and the DNA polymer is going to move from 5 prime to 3 prime direction, so we are not able to make this strand continuously, so we have to make it in a form of a small fragments that they call them Okazaki fragments. And eventually these fragments, with the help of some enzymes like a ligase, they're gonna attach together and make a complete strand. But since we make this strand in a small fragments and uncontinuously, so we call this strand lagging strand. So leading strand, we make it continuously, but lagging strand, we make it uncontinuously. So, and if you look at the lagging strands, so after we made these Okazaki fragments with the help of DNA polymerase 1, we're gonna replace the RNA molecule with the DNA nucleotide, because these are all these like greenish part, these are small RNA, so we have to replace them with DNA with the help of DNA polymerase 1, and then with the help of ligase we're going to seal and glue them together, and then topoisomerase 4 going to help to release the new DNA molecule, which is like a double-stranded DNA, which has exactly the same genetic material of the original DNA, will be released. So this is a very nice summary table for you guys. You can see that you can see the important enzymes and their functions in prokaryotic cells. And eukaryotic cells, they're gonna have the same steps in DNA replication. There are some differences, as you can imagine, because eukaryotic cells is much more complex. So in eukaryotic cells we have different origins of the replication, since it is they have a bigger and more complex DNA. So we have several origin of replications that they can do the DNA replication simultaneously at the same time. And another part that you can see in the eukaryotic cells, we call it the telomere. So telomeres are like the end of the DNA molecule that because of the way that the DNA replication works, sometimes it can cause that the new strand be a bit shorter than the original strand or the parental strand of the DNA that we have. So with the help of the enzyme that we call it telomerase, so we can make sure that the end of the linear chromosomes are going to maintain by the action of this enzyme, and we are not going to have a shorter DNA. And it's a very important topic in cancer studies, in stem cells, aging, and very, very important topic that a lot of labs they work actually on it. So, and Elizabeth Blackburn, she was actually the person that she discovered how telomerase works, and she received a Nobel Prize in 2009 for her discovery. So these are some of the enzymes that you can see in the DNA replication in eukaryotic cells and prokaryotic cells, and some of the differences that you have we have between these two types of cells. So like origin of replication, we have a single origin in prokaryotic cells, we have multiples in eukaryotic cells, and the rate of the replication is much slower in eukaryotic cells compared with prokaryotic cells. We have different types of DNA polymerase that we talk about some of them. There is no telomerase in prokaryotic cells, and they have different enzymes, like but they might have same like functions. And then you can see like some of these enzymes that vary, but overall the way that or the steps for making the DNA replication in prokaryotic cells and eukaryotic cells are very similar. So, and if you remember, we talked about plus those chromosomal DNA that we have some other DNAs in the cells. For example, in bacteria, we have those plasmids, those circular DNA, small DNA, or bacteriophages or animal viruses. Some of them they have circular DNA. So this DNA, in order to make the replication, so they can do the replication, for example, in the plasmid completely separately from the genomics DNA, and this plasmid, so they gonna, or these circular DNA viruses, they can actually use rolling circle replication to make the new strands. So they can make single-stranded DNA, and then they also can make double-stranded DNA as well. So usually it's unidirectional because it's in a circle, so it goes in one direction, and then they can make a new plasmid, or they can make a new DNA in that virus. So we're going to continue about the next role of the DNA, that we know it as the blueprint, in the next video. Thank you very much, and talk to you soon.